So the structure of the dipeptide thyrolu is shown. The peptide bond is a covalent bond between the carboxyl group of threonine and the amino group of leucine, and an N-terminal is the end of the peptide chain that has a free amino group, while the C-terminus is the end that has a free carboxyl group. It's the N-terminal, carboxyl, and then the amino terminal. The peptide bond and then the N-terminus and the C-terminus would be in the format of that. And the protein structure can be classified into 4 levels: a primary structure, a secondary structure, which the primary structure is a linear sequence of amino acids in a polypeptide chain. It is determined by the genetic code and is held together by the covalent peptide bond. For example, the primary structure of insulin is shown below, which is an insulin A chain and then an insulin B chain. The secondary structure is a local folding of the polypeptide chain into regular structures such as alpha helices and beta sheets. It is stabilized by hydrogen bonds between the backbone atoms of the peptide chain. So, for example, the secondary structure of myoglobin, a protein that binds oxygen in muscle cells, is shown below. Tertiary structure is the overall three-dimensional shape of the polypeptide chain resulting from various interactions between the side chains of the amino acids. These interactions include ionic bonds, hydrogen bonds, disulfide bridges, hydrophobic interactions, and van der Waals forces. For example, the tertiary structure of the myoglobin is shown. Quaternary structure is an arrangement of multiple polypeptide chains into a functional protein complex. It is stabilized by the same types of interactions as the tertiary structure. Not all proteins have the quaternary structure, though, as some proteins consist of only one polypeptide chain. So, for example, the quaternary structure of the hemoglobin, a protein that carries oxygen, is in the blood, is shown. Hemoglobin consists of the polypeptide chains, 2 alpha chains and 2 beta chains. Enzymes are a protein catalyst that increase reaction rates by lowering the activation energy. Activation energy is the amount of energy required to reach the transition state, an unstable state that products must pass through in order to become reactants. Enzymes lower the activation energy by binding to the substrate molecules and facilitating their conversion to products. There are 2 types of enzyme substrate interactions that lower activation energy, which is an induced fit. This is when the enzyme changes its shape slightly to fit more snugly around the substrate after binding. This puts stress on the substrate and weakens its bonds, making it easier to break or modify. For example, hexokinase is an enzyme that catalyzes the phosphorylation of glucose. When glucose binds to hexokinase, the enzyme undergoes a confrontational change that closes its active site around the substrate and includes water molecules. This induces a strain on the glucose molecule and facilitates the transfer of phosphate group from ATP. Proximity and orientation. This is when the enzyme brings the substrate molecules closer together and aligns them from favorable position for the reaction to occur. This increases the probability of success collision and reduces the entropy of system. For example, DNA polymerase is an enzyme that catalyzes the synthesis of DNA. It binds the template strand of DNA and in the incoming nucleotide and positions them in the correct orientation for the formation of polyvadicere bond.